Hello friends, welcome to B.Sc.Agriculture. If you are coming to my channel for the first time, then don't forget to hit the subscribe button. Your subscription and likes are always motivating us. So if you have any doubts, please comment in the section. So let's move to the video. In this video, we are going to see about fundamentals of pathology. I am going to cover the role of enzymes in plant disease developments. So while seeing about them, you must need to know about the layers of plant. So the plant leaf layers are said to be made up of different parts like cuticle, upper epidermis, plasmid mesophilum, xylem, phloem, guard cells, stomata, cuticle. So even they used to have veins present in them. So there are different components which are said to be present in leaf layers of plant leaf. So while seeing about the layers of plant stem, so the primary growth which are said to be in plant leaf structures are phloem, xylem and sclerenchyma. So around them there is a secondary growth, so where you can able to see a secondary phloem, secondary primary phloem, cork, bark, cortex, secondary xylem and primary xylem around the layers. So I have given the picture of the layers of plant root also, so you can able to see the structures which are present in them as xylem, cortex, subraised endodermis, Casper strips, etc. So after that there is the first part where the pathogen used to take is the breaching of cell wall. So the many plant pathogens which are mainly entered through wounds as well as natural openings, or they use it to travel from one plant to another plant through vectors. So some of the fungal pathogen which use to achieve entry by mechanical force or enzyme activity, or also the combination of both, which I have told in the before lectures. So the first one I am going to explain is breaching by mechanical force. So I have already told that the direct penetration process, they used to have the suspended pressure on the leaf surface. So the melanized aproria which used to adhere tightly on the surface with massive tubular pressure, they are developed. They used to enter into the plant leaf. Example, rice blast pathogens, Magnaportha grisae. So these are some of the examples of breaching by mechanical force. So they use it to enter into the leaf by giving an osmotic pressure as high as 8 millipascal, which is the pressure around 8 bars. So they used to give a pressure roughly of around 40 times the pressure of cot air. So for its surface the pressure can be, which is said to be higher when compared to others. So I have also given another example of Plasmodium brassicae, which used to cause clubroot of cabbage. So I have given it as the systematic diagram of how the zoospore used to penetrate the host cell. So which is a I have also given another example, Loranthus hostorial penetration of the host one. So after that we are going to see about breaching by enzymes. So it is what the lecture mainly composed of. The breaching by enzymes, where the enzymes which is said to be known as the large protein molecule, which used to interrelate the reaction between the living cells. So these enzymes which are used to degrade the surface layer of plants like wax, cuticle, cork, epidermal layers. So the enzymes which are used by pathogens are lipolytic enzymes, cutinolytic enzymes, pectinolytic, cellulase, xylase, lignase, protease, etc. So these are the enzymes which are said to be produced by the pathogen, and they can able to easily enter into the plant cells. So it is the picture of cuticle, where you can able to see a waxy outer layer which prevents the entry of water from leaving the cells, as well as found only on the top side of the leaves, so the cuticular layer. So the lipolytic enzyme or lipase, so which is said to be produced by the pathogen and which used to degrade the wax layer surround the leaf. So some of the example are Bulmeria graminis, which used to germinate on the surface of barley and erode the surface wax of the barley leaves. So these are some of the pathogens which used to eradicate as well as erode the wax surface on the leaves. And after that I am going to tell you about the cutinolytic enzyme or cutinase. So which is said to be used by the pathogen for the degradation of cuticle. So Example, the cuteness producing pathogens are Ascochyta rabi, which is used to secrete cuteness, and as well as Bumellaria graminis, which is said to be produce cuteness in barley. So while seeing about how they produce, the cuteness is said to be produced by a wide spread of plant pathogenic fungi, which is said to be consecutive as well as indicable among the plant pathogens. So while seeing about the cuteness, there is said to be two types of cuteness. The first one is cutin estrase, which is said to be used for the removal of ester bond in between the cuticle, as well as carboxycutin peroxide, so which is said to be used for hydrolysis of peroxide groups of cutin, which is used to result it as the dissolved where the cutin is converted as soluble in water. So the cuteness producing microorganisms are Ascochyta rabi, Penicillium, Coltotrichum duosporidus, which are the pathogens which use to produce enzymes to infect the host plants. And I have also given the pictures of cork, which is in. So the subaerians, which is said to be a waterproofing waxy substance, which is mainly found in the stem of plants. So the subaerian is said to be a major constituent of cork, so which is said to be a barrier to movement of water as well as solutes in the host plant. So the D-grade subaerian complex are said to be produced by the pathogen. Some of the examples are Fusarium solani as well as Armillaria melani. So they use it to convert the subaerian into alphatic monomers as well as p-nitrophenyl butyrate when they are entered into the host plant. So, and after that we are going to see the middle lamella calcium pectate. So these are the pictures. So the pectinolytic enzymes, which is said to be used to degrade the pectinase substances. So the pectin present as complicated heterogeneous structure in between them. So the linear chains of pectase or pectin by two process or hydrolysis pectate and pectin, the polyglucotinase as well as pectinase. So these are the enzymes which is used for the convert as well as hydrolysis of pectate as well as pectin. They are mainly the hydrolysis process taken place by these pathogens. So the pectinolytic enzymes, so there is mainly a beta elimination process that to be takes place, as well as exopolygonate used to attack the polymer chains at the end, given rise to the dimers. So as well as the endopolymerase, which used to attack the chains at middle, which used to produce the mixture of oligomers in the host plants. After that, the de-esterification process takes place, and the pectin methyl esterase, which is used to de-esterify the pectin, as well as polygalactonase, where the Agrobacterium vitis as well as Erwinia caratovora. So these are the pathogens which used to produce polygalactonase, as well as some of the pathogens like Penicillium, Expanium, Aspergillus flavus also used to produce the polygalactonase enzymes. After that, we are going to see about pectate lysase. So the pectate lysase producing enzymes are Vervinia, Chrysanthemi, Coltotrichum, Duosporidus, Nectaria, Hematococca. So while seeing about the action of pectinase on the enzyme, so they used to first attack on the mid portion as well as the middle of the cycle, so they can able to convert it into oligomers or dimers based on the point of attack. So after that, we are going to see about the cell wall cellulose. So the cellulase enzymes are, there are around 3 type of cellulase enzymes. The first one is C1 enzyme, which is used to cleavage the cross linkage between cellulose as well as the crystalline nature is said to be degraded in C1 enzymes. And in C2 enzyme, they use it to attack the naive cellulase, so they can able to break them as shorter chains as well as expose glucose chain in between them. So it's how they use it to occur. And after that, we are going to see the hemicellulase, so which is a complex mixture of polysaccharides. So they are said to be around xyloglucone, glucanose, galactoman, arabinogalactans are said to be present in hemicellulose. So the hemicellulase are the xylase, galactonase, glucanase, arabinase and mannose. So the other enzymes are said to be or even produced by the pathogen for the destruction of hemicellulase, and lignase as well as lignolytic enzymes, which is said to be used for the degradation of lignin, which is a phenolipid polymer. So they are mainly present in middle lamella as well as the secondary circulatory system and in fibers. So they are also even present in epidermal as well as hypodermal cells. So while seeing about the characteristics of lignin, they are more resistant to enzymatic degradation. So the lignin peroxide, which is used to cause oxidative depolymerization in lignins. So it's how the lignin is used to act on the lignin compound, and some of the examples are the white rot fungi, which used to degrade lignin in plant cells, and proteinase. So these are the used for the degradation of protein substances. So some of the organisms which used to degrade proteins are Magnaportha gryza, Xanthomonas axonopolis, Septoria nodum. So these are the proteinase producing pathogens. So I have given the lecture notes in description. If you have any doubt, please come to the section and join us in the Instagram page. So if you like the video, like it and share it with your friends, and subscribe to B.Sc. Agriculture and hit the bell icon to get regular updates. So thanks for watching the video. I'll catch you up in next videos.